and we all work tirelessly to assist the more than 1 million people with Parkinson's disease in the United States live life to the fullest. So welcome to Bob, who is watching from Olympia, and to Susanna, who is in Seattle. Welcome. APDA has a dual mission of providing help for today to those living with Parkinson's and hope for tomorrow by investing in critical research projects. And research is what we are here to talk about today. APDA has invested nearly $51 million into critical research since 1961. And the Northwest chapter is proud to be the sole funder of the Parkinson's Disease Registry, which helps speed up the research process by connecting researchers to willing participants. Keep an eye on your post-event email for more information about that registry and how to get involved. There is amazing research going on in the Northwest, and we hope through this series, you will learn more about the work being done, the people behind it, and how you can get involved. Now, before I introduce today's speakers, I want to thank the sponsors of this program. The gold sponsors are Kiora Kieran, Synovian, and Niagara Bottling. Silver sponsor is Amneal, and bronze sponsor, Boston Scientific. Their support of APDA and their value for educational programs like this is immensely appreciated. Thank you so much. Today's Passport to Research series is entitled Research on a Molecular Level, What's in the Pipeline? And I'd like to introduce and welcome you to two Parkinson's disease researchers who will join our discussion this evening. Dr. Vivek Unni and Dr. Ian Martin, both from Oregon Health Sciences University, located in Portland, Oregon. Dr. Martin is a neuroscientist dedicated to understanding what causes PD. He received his BS degree from King's College in London and PhD from Virginia Commonwealth University. In 2009, Dr. Martin joined Ted and Belina Dawson's laboratory at Johns Hopkins University, where he worked on molecular pathways underlying Parkinson's disease that are linked to mutations in LRRK2. In September 2015, he joined the Junger Center for Neurosciences Research at OHSU as Assistant Professor of Neurology. And in 2016, he received a research grant from APDA to support his work. His lab at OHSU studies how mutations in LRRK2 cause death of dopamine neurons, leading to Parkinson's disease. Dr. Unni is a physician scientist caring for people impacted by Parkinson's disease, as well as a scientist conducting basic PD research. Dr. Unni has undergraduate and master's degrees from Stanford University and earned his MD and PhD degrees from Columbia University. He completed his neurology residency and fellowship in movement disorders at the Harvard Partners Brigham and Women's Hospital and Massachusetts General Hospital programs. Before joining OHSU, Dr. Unni was an instructor in neurology at Harvard Medical School and a research fellow at Mass General. In 2010, Dr. Unni received the prestigious NIH K08 Career Development Award, and in 2016, he received the Katsias Fellowship, an APDA-funded career development award. He is currently associate professor in the Department of Neurology and the Junger Center for Neurosciences Research at OHSU, where his lab uses a unique imaging approach to examine the trafficking and metabolism of alpha-synuclein in the brain of live mice. And Dr. Unni is also on APDA's Scientific Advisory Board. Welcome, Dr. Martin and Dr. Unni. Now, before we get started with questions for Drs. Martin and Unni, if you have any questions, please submit them in the Q&A tab at the bottom of the screen. If you could not submit them in the chat, but submit them instead in the Q&A, that would make following our questions from the audience easier to follow for us. So thank you very much for that. So on that note, I want to welcome everybody who is joining us, and we're going to start off our session. So for our first question, our session tonight is entitled Research from a Molecular Level. Dr. Martin, could you explain what that means and perhaps help our audience understand how molecular research fits into the broader umbrella of PD research? How does it relate, for example, to research that might involve people with PD?
Absolutely. So really one of the major goals of molecular research is to get a biological window into the Parkinson's brain to understand what the molecular alterations are at the level of genes or proteins, so the basic building blocks of life, to try to see what alterations give rise to dysfunction and loss of the brain cells that cause Parkinson's disease. And of course, one of the main goals is to understand what the underlying mechanisms of disease are. So if we think about a particular genetic mutation that causes Parkinson's disease, how do we get from A to B? So how do we get from a genetic mutation to changes in the protein that it encodes that eventually promote the loss of dopamine neurons? But another equally, or perhaps more important goal of this type of research is to identify what the molecules are, the molecular roadmap, if you will, that underlies the disease, in order to try to find what we would call druggable targets that we can design therapeutic interventions toward, and then ultimately move from preclinical testing in cells or animals to the clinic for clinical trials.
Excellent. So basically what you're saying is, before anything can be tried in people, it's got to be discovered first in a molecular lab, just like the lab you run and Dr. Unni runs. So your work is really, really vital to get things started. So thank you very much for doing what you do. And now let's learn a little bit more from each of you about your research projects. So Dr. Unni, could you share with us generally what drew you to the world of Parkinson's disease research and how your lab runs? What sort of experiments do you do, and what sort of things are you looking for?
Yeah, so as you mentioned, I'm a neurologist and I see patients with Parkinson's disease. And I started out getting an MD PhD, so kind of a combined degree. I always knew I wanted to see patients and do research. But when I was doing my training in med school, I wasn't really sure what I was going to do or what I wanted to do. And I sort of had the classic conundrum that a lot of physician scientists do, which is you have to decide what you're going to do base your career on. And there's kind of two major categories of ways of doing it. Some people kind of pick the research project they find the most interesting to them, and then kind of develop a clinical career or expertise around that. I kind of decided to do the latter. I knew I was very interested in the brain. I knew I wanted to study the brain, but I wasn't really sure what I wanted to do. But over the course of my neurology residency, I realized I really liked taking care of Parkinson's patients. So I kind of decided I would focus my career on Parkinson's, and so I would gear my research towards that, because the research I had done before I did residency was actually not related to Parkinson's directly. So I kind of took that latter approach. I don't know why exactly. It's a hard question to answer, really, but I do enjoy taking care of Parkinson's patients a lot. And I learned that early on in my experiences as a resident. So then I based my kind of research career around that. And so the way my lab runs, we use this technique that you alluded to, which allows us to look in the brains of living mice. So it was an approach that kind of came out while I was in graduate school, and it was something I sort of became enamored with very early on, before I could actually do it myself. But people developed this kind of microscopy technique, which allowed you to anesthetize a mouse, do a surgery where you remove a flap of bone from the skull and replace it with a glass cover slip. And if you do the surgery well, the mouse can live its normal life like that. And now you've created this literal window onto the top of the brain, and you can look in the brain of living mice at individual neurons. And to me, that just blew my mind when they first showed that you could do that, because it was the first time where you could actually look at kind of a cellular level at what was happening in the brain of a living mammal. And so when I had made the decision that I wanted to kind of study Parkinson's disease, I tried to combine my, that interest with this idea of being able to look in the brains of living mice. And so luckily, where I was at Mass General, one of the leaders in the field in terms of using that approach to study diseases of the nervous system, specifically Alzheimer's disease, was there. His name is Brad Hyman. And so I joined his lab knowing that I wanted to kind of work on Parkinson's disease. So together we developed this approach where we would take that technique but apply it to a mouse model of Parkinson. So the way my lab runs is we do kind of a whole range of experiments now. At one level are these mouse experiments where we can look at the brains of living mice. But we're also focused, as Ian was alluding to, on kind of the molecular level too, because underlying the disease is really kind of the interaction of different molecules with each other, and something's going wrong at that level. So we kind of, in my lab, try and span that whole range from the molecule up to the actual living organism as a way to kind of model what's happening in Parkinson's and develop new treatments for it.
Fantastic. Dr. Martin, could you share with us what drew you to Parkinson's disease research and give us a general overview of what goes on in your lab?
Sure. So my background is actually more in the biology of aging. So throughout the course of my PhD, I was fascinated by, why do some people maintain robust health into late life while others develop age-related diseases? And moving through the course of that graduate career, I became more and more interested with diseases of the brain, age-related diseases of the brain, including Alzheimer's and Parkinson's. And then actually, as you mentioned in the opening, the postdoctoral mentor that I worked with, Ted Dawson, he came to my graduate institution and gave a talk, and I was just blown away. So I really kind of fell in love with the idea of doing basic research into Parkinson's disease and trying to work in his lab. And thankfully he took me. I joined his lab and started working on the LRRK2 gene. And that has really, created a flurry of genetic studies that we've been working on in that lab and continue to working on in my lab to try to understand how mutations that in the LRRK2 gene that cause Parkinson's disease affect the properties of the LRRK2 protein and how that underlies, the dysfunction and death of dopamine neurons. And there's a lot of excitement about LRRK2 in the field because not only do mutations in LRRK2 result in late onset Parkinson's disease that in most ways resembles idiopathic Parkinson's disease, which is by far the most common, which is not true of many other genetic mutations that have been linked to the disease. So LRRK2 has that going for it. It's also an enzyme. So LRRK2 is a type of enzyme called a kinase, and actually mutations in LRRK2 that cause Parkinson's disease seem to increase activity of the enzyme. And so there's a lot of excitement about how we might be able to intervene in cases where LRRK2 has been activated by the use of kinase inhibitors that can be given to patients. And I should also say that LRRK2 mutations are pretty much the most common mutations found in Parkinson's disease. So they're found both in familial forms, but also in idiopathic forms. And so there's potentially broad applicability to LRRK2 kinase inhibitors and other therapeutics that might come out of LRRK2 research.
Fantastic. So Dr. Unni, we have a question coming from Ellen from our audience about your thoughts on mannitol and whether that is something that you think may be a therapeutic option, and maybe tie that into your work on alpha-synuclein in general and how alpha-synuclein may be a key player in manipulating how Parkinson's disease is treated.
Yeah, no, that's a great question. I know there's a lot of interest in the field in mannitol, and there's, I think some interesting work in mouse models and rat models that it can potentially be beneficial. I give a little pause because unfortunately there are many treatments that have been tried in mice and rats that have worked spectacularly, but when they get tested in people, actually all of them so far, in terms of say slowing the disease or stopping the disease from getting worse, haven't worked. So, I think it's really too early to say. I mean, there is some interesting biology there in terms of how mannitol could potentially interact both with kind of the blood-brain barrier, which is kind of the protective mechanism that the brain has to keep things inside the brain that should be and keep things out of the brain that shouldn't be, and also directly with aggregation of specific proteins. So the protein that I'm very interested in work on is alpha-synuclein, which is the primary component of these Lewy bodies, which are the kind of pathological hallmark of Parkinson's disease. And there's some data that mannitol might be able to influence how that protein aggregates. So I think it's definitely very interesting, but still way too early to say it's useful. And mannitol is one of those agents that's actually used for other indications in people right now. And it's pretty easy to get actually even over the counter too. So it's the kind of thing I wouldn't recommend people take just because we don't know yet whether it really works or not, or if it could even be harmful in some cases. So.
All right, thank you for that answer. We have a question from Barbara, and this is for Ian. How many patients or what percentage of people with PD have LRRK2 mutations? And let's expand that question a little bit. So if we discover something in your lab that may help people with or may interact with LRRK2 in general, would that be a treatment for just with people with the mutation or or for everyone?
Those are some great questions. So within there's basically two kind of branches of Parkinson's disease cases. We tend to think about familial cases where there's clear segregation of a genetic mutation within a family. And within that branch of cases, LRRK2 doesn't account for the majority. It's actually probably about 10 to 15% of cases. But actually, when we open it up to idiopathic cases, which are 90% of all cases, depending on the population under study, LRRK2 mutations can be found in about 1 to 5% of those patients. So those numbers in terms of percentages sound a bit low, but actually, when you think about that in terms of absolute numbers of patients, it's a lot. And these are, when I'm talking about mutations, these are known pathogenic mutations. So where there's clearly a link between the mutation and disease, there may be other mutations or what we call polymorphisms, risk gene variants in LRRK2 that may increase a person's risk of developing Parkinson's disease. And we're learning more about that over time. So kind of getting back to what I was talking about earlier, that is why there's so much excitement over LRRK2, because there's evidence also in the idiopathic Parkinson's patient population that even in the absence of clear LRRK2 mutations, there may be activation of LRRK2 enzyme activity in the brain of those people. And so there's, cautious excitement, I would say, that anything that is developed to target LRRK2 kinase activity, to reduce LRRK2 kinase activity and normalize it within the cells of the brain, that could be clearly applicable to patients with LRRK2 mutations, could also potentially be, applicable to a much wider net of people. And right now, those kinase inhibitors are undergoing clinical trials. And so, they've moved through safety testing and will be tested in Parkinson's patients with LRRK2 mutations, I believe this year. So we should hopefully learn a lot more about the potential effectiveness and benefits of those kinase inhibitors in the next few years.
Fantastic. We have a question from Leanne, which actually could be answered by either of you. It is related to Parkinson's disease and genetics. If someone does not have a family history, are not aware of any mutation that they particularly have, but they do have Parkinson's disease, what would account for developing Parkinson's in that sort of person? Would that person also have a mutation that maybe they don't know about? Or we just don't think about genetics in a person like that? Who wants to answer that one?
I'm happy to.
Okay.
It's a great question. I would say that if you look at all people who walk into the clinic, even those who have no family history, if you do test them all, then as Ian mentioned, probably 5 or 10% of them will have a mutation, even though there's no clear family history. Now, if they have a clear family history, now having one affected first-degree relative, so one of your parents or one of your siblings or one of your children, that doesn't increase your risk that much more. But if you have 2 affected first-degree relatives, then actually your risk of having a mutation or a genetic form goes way up. Okay, so the people who, let's say, are in the bigger group, much bigger group, let's say the 95% of people who don't have a clear mutation that we know about, what causes their disease? And this is kind of one of the oldest questions in the field. And, the real answer is we're not sure. I would say that it's probably very likely, and this is my personal opinion, that the cause in those people is going to be some combination of an environmental exposure and inheriting a more complicated genetic susceptibility. So, you can think about genetics in different ways. There's kind of the simple genetics where you inherit one bad gene from one of your parents, and that gives you a disease. But, and in many diseases that we've studied, because they're easier to study, work that way. But in reality, genetics is much more complicated because you inherit, 20,000 genes, one copy of each from each of your parents. And so, having certain combination of the genes may not cause the disease by themselves, but when you have that certain combination and you're exposed to a certain thing in the environment, that could maybe trigger a disease. And, there's good evidence for other diseases that can happen. So I wouldn't be surprised if that's what's going on in many people who have, let's say, not a simple genetic form of Parkinson's. And, there are definitely environmental risk factors out there that have been, described and there's evidence for. So probably one of the ones that Ian is actually an expert in is kind of pesticides that are out there that are used in agriculture that can increase people's risk. There's evidence that head trauma previously in life can increase people's risk. It's hard to directly say in any one person whether they got the disease because of one of these environmental risk factors, but definitely if you look at a big group of people, if they do have this risk factor, they're at higher risk for developing Parkinson's.
Fantastic.
And I would just add to that, if I may. I mean, 25 years ago, it was controversial to say that genetics played a role in Parkinson's disease. Then in the late 90s, mutations in the gene that encodes for alpha-synuclein was linked to Parkinson's disease. And that's really ushered in the modern era of Parkinson's genetics. And now with modern genetic sequencing technology, we're able to take a much more nuanced approach to identifying genes that can be associated with Parkinson's. So moving beyond cases where there's clear segregation within a family of a particular mutation to examining the broader population as a whole and identifying subtle risk variants that increase somebody's risk of developing the disease at a small amount, and if they have enough of those risk variants, can potentially tip the scales to genetic predisposition. But as Vivek mentioned, it's the old saying that it's genetics that loads the gun and environment that pulls the trigger. And these are genetic predispositions, and there's also likely very important environmental factors that also determine whether somebody would or would not develop the disease.
Great, very well said. Thank you. I have a question that, again, either of you could answer. There has been a tremendous amount of research in Parkinson's disease over the last number of years. Would you say that compared to, let's say, 5 to 10 years ago, are we closer now to a major breakthrough in Parkinson's disease treatment and cure than we were? Do you have any sense of whether this is a time for hope or whether this is sort of more of the same?
I could start off by answering that one. I no doubt have hope. The big question is when. I get asked this question all the time by my patients in clinic. When is there going to be a big breakthrough? And I don't have a good answer for that. And what I usually try and explain is it's sort of like predicting an earthquake in many ways. I can tell you in the next 50 years there's probably going to be a category 8 or let's say 7.5 earthquake somewhere in the world, and I can't tell you when, and I can't really even tell you where, but I can tell you where things are much more likely to happen than not. And so I kind of see predicting science in the same way. Like I have a lot of confidence that in a period of time we're going to make major breakthroughs. But is that earthquake going to happen tomorrow, or is it going to happen in 5 years, or is it going to happen in 10 years? And is it going to come from studying LRRK2, or is it going to come from studying synuclein, or is it going to come from studying something else, I can't really give you an honest answer. But I do think, LRRK2 and synuclein and some other topics are kind of the most likely places where, breakthroughs are going to happen. And I think we've learned a lot about these molecules in the last 20 years. So I think our chances of, making big discoveries are going way up. But unfortunately, I can't give like an answer that, I think in 5 years for sure, we're going to have a drug that, stops the progression of Parkinson's disease. Just because the predicting science, I do think, is a lot like trying to predict an earthquake.
Thank you for that very honest answer. Dr. Martin, you want to add anything to that?
Yeah, I would also say I'm optimistic. The pace of discovery is accelerating, and this is partly due to just modern technology that feeds into what we're able to see and do with our disease models and what we're able to accomplish on a given day in the lab. But also just in the appreciation for the importance of diseases like Parkinson's disease. We have an aging population, and so if 1% of the population over 60 to 65 is going to develop Parkinson's disease, this is only going to increase with time. And I think the recognition from governments and thought leaders, of the importance of these problems is certainly funneling a lot more money into Parkinson's research and Alzheimer's research. So for those reasons, I remain optimistic. But as Vivek said, trying to pinpoint when or how it's going to happen is kind of hard.
Fair enough. We have some excellent questions rolling in. This is one for Dr. Unni from Mark. The supposed central role of alpha-synuclein has been suspected for over 20 years, and a number of trials and research have been focused on some aspect of this protein. However, it is still not known whether the misfolding is a cause or a result of Parkinson's. Can you address that?
Yeah, no, I agree with everything that was just said in that great question. So it is clear that Parkinson's is related to alpha-synuclein and alpha-synuclein aggregation. Actually, by definition, to be definitively diagnosed with Parkinson's disease, the way our gold standard is that at autopsy, there has to be aggregates of alpha-synuclein in the brain. But whether that's really what's driving the disease or not is, I think, still a very open question. I mean, I've sort of bet my career that it is, because and there are reasons why. I think there are other diseases where you see alpha-synuclein aggregation and you also get kind of similar patterns of neurodegeneration and disease, and dementia with Lewy bodies is an example that people may have heard of. So I think there are multiple diseases now that are linked to aggregates of alpha-synuclein, but that doesn't definitively say that it's the aggregate itself that's causing the disease. But one of the things I think that's really inspired me is that all this research that's been happening over the last 10 or 20 years, we have much better models now of Parkinson's disease in, let's say, animals like mice than we ever did before, and specifically models of the alpha-synuclein aggregation. And that's really only been around for less than 10 years that we've had those models. And so, I think if alpha-synuclein is related, we can actually now test that, because before 10 years ago, you couldn't really make a Lewy body model in an animal or even in a cell in a dish. And so if you think synuclein is one of the big factors, then how do you study that and make a drug that changes that? Because the model wasn't really even around. But like I said, the last 10 years have really changed that. And so I think it gives us, this great opportunity, and myself, great hope that now that we can, see these alpha-synuclein aggregates in these model systems, now we can really try and attack them and see if we can do something. And that will really answer the question, right? Because if we can make alpha-synuclein aggregates go away in a mouse with a certain drug, then we can test that in people. And if that makes alpha-synuclein aggregates in people go away, and importantly, if that makes people better, then that will be ultimately what will tell us the answer to that great question.
Fantastic. Thank you. We have two excellent questions from Barbara, both on elements of genetics. So I'll ask Dr. Martin to answer these. The first is, do different mutations cause different symptoms in Parkinson's disease? Are there variants of PD based on the mutation? Great question. And the other question that Barbara has is, do you think that CRISPR technology is in the future of Parkinson's disease research and treatment?
So definitely mutations in different genes can produce different pathological outcomes and different symptoms in the clinic. But within a particular gene or protein, it's very hard to say if, if we can see differences at the level of symptoms. We don't really have the information or the data, I think, to make those conclusions. Ultimately, one of the limitations is that the number of available samples or patient samples or patients that are seen in clinics that we could try to draw those correlations from is too limited. It's not enough. And then as to the second question, absolutely, I think CRISPR technology is revolutionizing science.
Maybe just give a brief introduction of what CRISPR technology is for those listening who may not.
So it's a way to basically go in with kind of precision cutting tools and manipulate the genome, which is basically the cumulative DNA of an organism. And so we do this a lot now in the lab in disease models. So in cells or in animals that we want to engineer particular mutations into to ask particular questions like, can we, if we see a mutation that is found in a particular patient population, if we introduce that into an animal model, what happens? What can we learn from that? But it's also used to correct mutations. So one of the really new exciting kind of frontiers is to take what are called pluripotent stem cells that are derived from Parkinson's patients that have known genetic mutations, and then try to correct those mutations in those cells. And then we can actually transform those cells down the pathway of becoming neurons. So we can make them into dopamine neurons and just see whether correcting those mutations in those cells is enough by itself to remedy the effects of those mutations on cells. And that's obviously a long way from understanding how those corrections may, how the results of those corrections in patients. But it kind of begins us on the road to that discovery, and is a really exciting new area of science.
Fantastic, thank you. We have a question about alpha-synuclein from Jennifer for Dr. Unni. Does alpha-synuclein research show that PD might begin in the gut? If you could address that.
Another great question.
Great question.
A lot of great ones today. That's very impressive. So yes, that is one of the kind of leading hypotheses out there. It's one that's been around for, I would say, maybe, I think the first paper suggesting it came out in 2003, so maybe 18 years ago. And it was around for a long time, let's say the last 18 years, but the evidence for it maybe being correct has kind of accelerated, I'd say, in the last 5 or 6 years. Now that doesn't mean it's still correct, but maybe I'll just review kind of what I mean to give people some framework for understanding. So there's this idea that, and coming back to alpha-synuclein, that that we know that when the aggregates of alpha-synuclein, the Lewy bodies, are in the dopamine neurons in the midbrain, those dopamine neurons die, and that causes the motor symptoms of Parkinson's disease. But what's also clear is that many patients, when they develop the motor symptoms of Parkinson's, have had other symptoms for many years. And it seems like those symptoms are actually part of Parkinson's, and they're evidence that maybe Parkinson's began years or decades before they actually developed the movement problems that led to their diagnosis. So some of the classic ones that happen very early are constipation, so a problem with the gut, and gut motility, and loss of sense of smell. Those are probably the two earliest ones. Now, not every Parkinson's patient has these problems early, but definitely you see this a lot more than you would expect, that people can sometimes have constipation 10 or 20 years before they develop the motor symptoms of Parkinson's. And so that led, along with some pathology work, which showed that you can get alpha-synuclein aggregates both in the olfactory system, which allows us to smell, and also in the nervous system of the gut, which controls the gut and gut motility. And when that goes wrong, it's easy to think how that could lead to constipation. People had shown that. That's where this idea came from back in 2003. But really what's been, catalyzing that idea or pushing it even more forward is recently is that now we can sort of see better evidence of that happening. Some of that comes from these models I was talking about, where you can get, you can start alpha-synuclein aggregation in the gut and then see it go into the brain, which is sort of, the idea that we think might be happening in people. And I want to say that it's definitely not established that this is the case. There's some people who would say that actually what's more likely is that Parkinson's disease begins in the brain and then it can spread down into the gut, and then people can get constipation later. And actually that you do see that. We do have patients who have the motor symptoms first and constipation later. So I think it's still a very open question about how this is happening, but I do think it's definitely, if you polled most, well, I mean, maybe that's the wrong way of putting it, but let's say many scientists out there who study Parkinson's think that that's what's happening. And if you force me to put my money down, I would say I probably believe that hypothesis as well. Although I do think there may be multiple places where it can start, that it's not always in the gut. It's maybe sometimes it can start other places, but I wouldn't be surprised if some people it actually starts in the gut and spreads into the brain.
Fantastic, thank you. I think we probably have time for just one more question. And this is a question, another great question from Bob. And this would be for Dr. Martin. Is there a greater potential for more severe PD symptoms from a known genetic cause of Parkinson's or an environmental exposure? How would you approach that question?
It really depends on the individual gene or exposure. It's not a one size fits all. So there are some genetic mutations that are completely penetrant. So in every individual that has them is going to develop Parkinson's disease. Clinically diagnosed Parkinson's disease. And there are other mutations that may not cause clinically diagnosable Parkinson's disease. There are certain environmental exposures that are very detrimental, that can cause acute onset Parkinson's disease. And one of those would be exposure to the compound MPTP. But then there are other compounds that are found in the environment, pesticides, heavy metals, pollutants, where the evidence linking to Parkinson's disease is not as clear. And certainly the severity of symptoms, is hard to kind of say one way or the other that environmental causes or genetic causes are, more or less severe.
Wow, everything just leads to more and more complication. The brain is really fascinating and full of surprises, that's for sure. I want to take this opportunity to thank Drs. Martin and Unni for joining us tonight and for your exceptional insights and your very clear way of explaining research and what you do. So thank you very much for being here on behalf of APDA. And thank you everybody for joining us and listening and contributing the wonderful questions that you did. If you would like to watch this again, or if you would like this to share this with anyone, later this week, it will be posted on the APDA Northwest YouTube channel. So please check that out and view it and share it. And we hope that you will join us next week, and hopefully Dave Grosby will be back, as we will speak with Dr. Jason Aldridge about novel trials with novel mechanisms. So to be continued, this wonderful series next week. And on that note, I hope everyone has a wonderful evening, and we hope to see you at another APD event very, very soon. Good night, everyone.